In this video presentation, I'll discuss one by one the factors affecting the action of disinfectants. But first, let's define what is disinfectants. Disinfectants are chemical compounds used to destroy and inactivate microorganisms on inert surfaces like benches, tables, and other surfaces. They are also known for their antimicrobial abilities. Despite this, they do not kill spore-forming organisms and can only disrupt their vegetative forms. The first factor affecting the action of disinfectants are their types and concentration. Disinfectants are categorized into two broad types: oxidizing and non-oxidizing agents. Oxidizing disinfectants are chemical agents that destroy the organism's cell membrane, leading to the impairment of their cell's ability to function by the process called oxidation. This type of disinfectant includes halogens, chlorine, iodine, bromine, and chlorine dioxide, as well as oxygen-releasing materials such as peracetic acid and hydrogen peroxide. On the other hand, non-oxidizing disinfectants can penetrate through the organism's cell wall and disrupt the phospholipid molecules that make up the cell membrane, thus blocking the metabolic pathways, causing the organisms to burst out their vital organs and die. So unlike the oxidizing disinfectant, this does not undergo the process of oxidation. Examples of these agents are quaternary ammonium compounds, amphoteric, biguanides, and acid anionics. Concentration will also affect the effectiveness of the disinfectant against microorganisms. The graph here on the right side shows the effect of disinfectant concentration and contact time on mortality of typical microorganisms. The y-axis here shows the concentration of microorganisms in log units or ml, while the x-axis here represents the time in minutes. The declining lines here also represent the concentration of the disinfectant, while this area here indicates the initial numbers or value of microorganisms. In interpreting this, as time passes after applying the disinfectant, the concentration of the microorganisms decreases, and the higher the concentration of disinfectant, the lower time it takes to kill or decrease the concentration of microorganisms. Basically, as the concentration of the disinfectant is increased, the rate of kill of microorganisms also is increased. The next factor is the type of organism. The table on the right shows the rank of organisms that are resistant to most disinfectants, with the prions as the most resistant of them all. Prions are infectious proteinaceous agents that are formed due to the abnormal folding of the proteins in the brain. They can also resist the regular heating temperature of the autoclave, which is at 121 degrees Celsius. Next, bacterial spores are dormant type organisms that have minimal metabolism and respiration. These organisms can survive harsh conditions and environments, all thanks to their spore-forming ability and other mechanism skills. They are also resistant to most disinfectants. On the other hand, mycobacteria are also resistant to alcohols. These organisms have a thick layer of peptidoglycan around the cell membrane made up of proteins and lipids. And finally, the non-lipid and lipid fungi, viruses, and bacteria are known for their resistance to most common or low-level disinfectants. We also need to consider the number of organisms. The graph here on the bottom part shows the effects of exposure time versus the number of organisms, where the y-axis here shows the log number of organisms, while the x-axis here shows exposure time in minutes. In interpreting this, the larger the number of microorganisms, The longer the time or exposure the disinfectant needs to kill 99.9% of organisms on a material surface. Another factor is the temperature. Most disinfectants' antimicrobial activity increases as the temperature increases, but some have exceptions. It can also degrade and weaken the action of disinfectant by increasing its temperature way too much than its normal degree, thus producing a potential health hazard. So, it is typical that disinfectants are used at a room temperature, about 20 to 22 degrees Celsius. In pH solutions, some disinfectants are active at only specific pH. Thus, an increase in pH enhances the antimicrobial activity of some disinfectants while others may decrease. The pH also influences the action of disinfectants by altering their chemical molecule or the organism's cell surface. Presence of organic matter. Organic material or matter in the form of blood, pus, feces, or lubricant material can inactivate or intervene with the germicidal action of disinfectants such as bleach, by the process called chemical reaction. Additionally, it also prevents full contact with the agent, thus protecting the organisms by acting as a physical barrier from any attack. We also need to consider the length of exposure of organisms to certain germicide. Materials must be exposed to the disinfectants for the appropriate minimum contact time. For example, it would only take 1 to 2 minutes for alcohol and iodine to destroy certain microorganisms. Endospores, on the other hand, are resistant to disinfectants, so autoclaving is needed to eliminate these microbes. And lastly, pressure applied or gaseous disinfectants. The relative humidity is the most significant factor influencing the activity of gaseous disinfectants such as chlorine dioxide, formaldehyde, and more.